Hi, everyone. It's such a pleasure to welcome you to today's Facebook Live discussion in partnership with the RET-positive patient group. For those of you who don't know me, my name is Dr. Rupal Basuroy, and I am the Executive Director of Research at LUNGevity Foundation. And this discussion is on a project that the RET-positive patient group is funding in partnership with LUNGevity Foundation. And before I introduce our very, very exciting panelists, and before we get to chat about the amazing science that Dr. Drake is going to be talking about, I wanted to share a couple of housekeeping things with all of you. The first one is, since this is a Facebook Live discussion, please use the Facebook chat function to comment or ask questions to our panelists, and we'll be monitoring the chat and we will be asking questions as they come in. The other piece is that this is being recorded, so if by any chance you miss some of the discussion, you've got to step away, that's totally fine. You can come back and watch it. And of course, this is purely for informational purposes. This discussion is not meant to be used as any information that should be used for medical decision making. So if you learn something that you're curious about today, please feel free to chat about it with your doctor. And last but not least, one of our panelists, Dr. Hilary Hamill, who most of you know, she's unable to join us today. She is recuperating from treatment, but she sends her very, very best to all of you. So thank you again for joining us. And now I am going to ask our panelists to introduce themselves. I would like to start with you, Ms. Walthall.
Okay, as many of you know, I'm Emily Walthall, and I am a stage 4 RET lung cancer survivor. I was diagnosed a little over 3 years ago with RET lung cancer, and I've been on Retevmo, my first line treatment, ever since.
Thank you, Ms. Walthall. Now I'm going to ask Dr. Justin Drake to introduce himself.
Yeah, hey everybody. I'm Justin. So I'm associate professor in the Department of Pharmacology at the University of Minnesota Twin Cities. And full disclosure, I'm also chief science officer of a startup company in the Twin Cities area known as Astron Biosciences. And my research focuses a lot in cancer therapeutics, and in this case, we're going to talk a lot about RET and its role in lung cancer.
Thank you again, Dr. Drake, for joining us. And Dr. Drake is the 2024 RET-positive Research Awardee. And I want to remind everyone that the RET-positive group is a pan-cancer group. It
Yes.
includes all patients diagnosed with some kind of RET-positive cancer, for example, lung cancer, thyroid cancer. So with that, Emily, let's start with you. You shared a little bit about your experience with getting diagnosed with RET-positive lung cancer. How did you find the RET-positive group? Why did you get involved with lung cancer advocacy?
I found the group through my doctor at UNC. I'm here in North Carolina, my home state. And when I was diagnosed at age 39, I was, of course, in total shock. And I didn't know what I wanted to do with all that I was feeling in terms of the emotional weight. And my doctor shared about RET positive with me. And UNC is one of 3 comprehensive cancer centers in North Carolina. And when I found out at age 39, again, as a never smoker, that I had lung cancer, I knew I needed to get to a research hospital, and that research was critical. As everyone here on Retevmo probably knows, this drug was only FDA approved in 2020. So, the role of research is so critical, and I knew I wanted to be part of a group advocating for lung cancer and other cancer patients like myself. And I also really value the support piece, but the research for me personally, and I know I speak for Hillary as well, really is at the forefront of our concerns as cancer patients with metastatic disease.
Thanks, Emily. Such an important point, right? Here are patients and caregivers who are invested in driving research that may change the disease outcome that they're going through. This is incredibly powerful, and this is something we did not have even 10 years ago. Thank you again, Emily, and the RET-positive group for everything that you're doing. Dr. Drake, let's talk a little bit of science now, and let's start really, really broadly, and let's talk a little bit about what your lab focuses on.
Yeah, so my lab at the University of Minnesota is primarily a prostate cancer lab. We are branching out mainly because of this RET connection, and I'll get into that in a little bit. But we have kind of two main buckets in the lab. One is to look at proteomics, which is another omics. So a lot of cancers, patients diagnosed with cancer that have RET fusions or RET mutations typically are found through what's known as genomics. You look at your DNA and you look for mutations in that front. DNA, RNA, protein, we look at the proteins and we try to see, are the proteins also made and are they also mutated, and they could be drivers of disease as well. So that kind of plays a role into this a little bit. And then on the other bucket is trying to understand the role that RET plays in various cancers and how we can go and treat that RET dependent cancer. And there are two approved therapies that are actually very, very powerful right now for RET positive disease. But there are some cancers that don't have these RET mutations at the DNA level. And we're looking at it at the protein level to see is RET indeed activated, even though you may not find those mutations. And we're trying to understand why that may be the case. So that's kind of the main things that we're doing currently in the lab.
That's fantastic, Dr. Drake, and thank you for reminding us that that sort of sequence of events, DNA, RNA, and then protein, and really appreciate the fact that you're focusing on looking at proteins and how how proteins can be targeted for cancer therapy. And you also mentioned that you're looking at all types of RET cancers, which is, again, great, and that very much aligns with the mission of the RET-positive group. So, Emily, tell us a little bit about why the RET-positive group selected Dr. Drake's research project to be funded. What were you most excited about? I know you're a part of the selection process.
Well, as a non-scientist lawyer, I will say what I can. I was very grateful to be included in the discussions and evaluations, which were very important to us as patient advocates. And, I think Dr. Drake's research is very exciting for us, number one, because of the pan-cancer approval. Our founder, Aaliyah Bricker, it was part of Aaliyah's vision that, all RET-driven cancers need a space. We have people in our community also with thyroid cancers, several pancreatic cancer patients. I think we have one woman whose husband has salivary gland cancer, but RET can drive a whole range as an oncogene of disease, and we didn't want to be exclusively focused on a particular tissue of origin. So, and I think the other thing for me is, again, as a non-scientist explaining my view about what Dr. Drake is doing, it's exciting in terms of the potential to prolong response. We know that these medicines, Retevmo and Gavreto, are incredible. They're very well tolerated. They're generally very durable in terms of response, but they're not cures. So duration of progression-free response is critical to our community. And I also think it's important how these protein degraders, this type of medical advancement is not tied to having necessarily a specific alteration, but so you're expanding the impact and potential to benefit more cancer patients.
That's such a fantastic point you make, Emily, about the broad range of impact a project such as Dr. Drake's can have. And I'm going to sort of pick up on that thread, Dr. Drake, and you brought up the two drugs, selpercatinib and pralsetinib, and you made a point that these two drugs work in, or they're used for a subset of RET-positive RET-positive cancers, but not all RET cancers. I think, for the RET community, people are interested in two questions, right? For those who can get pralsetinib or selpercatinib, what's next if the treatment starts stops working. And for those who don't have any options, and to your point, those who cannot get selpercatinib or pralsetinib, what options do I have? So can you talk a little bit about how your project is sort of chipping away at these two broad questions?
Yeah, for sure. And first, Emily, for you claiming you're a non-scientist, you do a great job of explaining things. So,
She also
keep
asks
that
hard
up. Yeah,
questions. She's
that's
the
amazing.
lawyer.
Um, so obviously with any targeted therapy, especially towards kinases, which is what RET is, you do develop, you do have resistance mechanisms that develop. Single agent drugs, that's typically true. This is not just a lung cancer or thyroid cancer problem. This is a pan cancer problem. So this happens in a lot of different cancers where you even have a targeted agent that may work for a while. It may ultimately not work somewhere down the road. All right. And the question is what's next? What do you do? How do you think about it? And there's a few things that we think about as scientists when we see this. So one is, does the target that we're going after, does it change? Okay. So in other words, does it create new mutations that may bypass that drug's efficacy? Which in this case, in RET-positive cancers, that is indeed one mechanism of treatment resistance, is that new mutations in the RET gene happen that make selpercatinib or pralsetinib ineffective. All right, these are called RET solvent front mutations, which can then drive the tumor to keep going, even though the drug may be efficacious early on. There's also what's known as bypass mechanisms of resistance. So there's other pathways that could get turned on that may drive the disease somewhat independent of the target you're actually trying to treat. So in this case, if you're giving selpercatinib to RET, and it may work, but what may happen is the cell may adapt and then turn on other pathways where selpercatinib may not be as effective because the MAPK pathway, which is downstream of RET, may become activated, or you may get a genomic amplification, which means more copies of a gene of another gene that drives a disease like MET, which is another receptor tyrosine kinase, MET. So these are different things that can happen in the cell and can drive treatment resistance. And so we're trying to understand how those happen, and then B, can we develop new therapeutics that go after those new mutations or those bypass pathways to potentially give in sequence or even potentially give up front as combination strategies to prevent that treatment resistance from occurring in the first place. So those are some of the typical mechanisms that happen in that context. And we'll probably get into the degraders here in a minute, but that's why I feel that the protein degraders, that's part of the project that RET-positive and LUNGevity have helped fund, helps to potentially eliminate some of those resistance mechanisms by going after the protein itself, independent of the mutations that could occur with the given therapeutics that are administered.
And I think that's what excites us as well. And before I ask you the next question, we're going to get really deep into biology. I did want to put in a plug for making sure that if your cancer progresses or comes back, then please ask your doctor to do a rebiopsy at progression and make sure you get that testing so you can understand what is driving that growth. It could be, as Dr. Drake pointed out, new mutations on the RET fusion itself, or it could be new mutations or amplifications to other genes such as MET that Dr. Drake was talking about. The piece, again, I'm going to go back to protein. You mentioned that these new classes of drugs, they target proteins. And right now, the two drugs that we're talking about, selpercatinib and pralsetinib, they belong to a class of drugs called tyrosine kinase inhibitors. And your drug class is very different, Dr. Drake. So can you talk a little bit about what is a protein? Let's start very basic. And why do we care about proteins and protein degradation?
Yeah, absolutely. So, the central dogma in science is DNA, RNA, protein. I've said this, a little earlier in our discussion. And, proteins are the functional unit of a cell. They drive all the biology of the cell. So they're actually critically important. If you don't have proteins, you're not who you are. You're not going to exist. So proteins are very important in that context. And most drug therapeutics go after proteins. Very few of them go after the DNA, and very few of them go after the RNA. So you really need to understand what's going on at the protein level to understand how these drugs are actually working, in the context of treating a patient for a given cancer. So that's kind of the focus and kind of how I have always thought about it. The field of proteomics, I think, is moving in that direction. And I do think in the next 10 years, we're going to see a big increase in the use of proteomics in the context of treating patients with cancer by understanding what proteins are expressed. Maybe we can identify the mutant proteins as well, but we can also find the activity of those proteins. Okay, activation of those proteins is also important, because if a protein becomes activated, it can essentially then turn on downstream proteins or proteins down below it in a cascade that could drive the growth of that cell. All right, so if you can stop that cascade, you can then essentially, kill that cell or deem that cell, more moot. All right, so that's kind of how we think about it from that context. So, that's kind of a long answer to what a protein is, but I think you have to think of it in that broader context to really understand why we think proteins are really important. And then, the class of therapeutics that we're looking at are known as protein degraders. So again, it's the concept and theme around proteins. But instead of using an inhibitor, like a kinase inhibitor, to block the activity of that protein, we're actually going to go in and eliminate the protein altogether. So we're going to wipe it out. It's not going to be around anymore. And there are mechanisms in the cell that can actually do this. It's called the proteasome, so the ubiquitin proteasome mechanism. So a lot of proteins in your cell are actually, targeted for this every minute of every day. So it's not like this is something that's unique to what we're trying to do. We're just hijacking that system in the cell to use it for, our advantage, which would be to treat the cancer itself. So what we can do then is we can use a compound like a kinase inhibitor. So we could use selpercatinib as an example, which we know binds to RET. All right, we know where it binds, we know its function. And then we can add a little linker to that compound, all right, which is just a little chain of carbons, and then put on a degrader on the other end, which would be kind of a ubiquitin ligase, all right? So that ubiquitin ligase, what that does is that'll go in and then it'll add ubiquitin proteins to the RET protein itself, all right? When a protein has ubiquitin on it, it targets it for degradation and it gets chopped up into little pieces and gets recycled. So that's essentially what the function of these protein degraders are to do, is to eliminate the protein using that mechanism. So, we're very excited about taking this new approach, and we can talk about the advantages of why we think degraders could work in that context if you'd like to. But we do think they have a lot of advantages over what the existing kinase inhibitors can provide. And, on a kind of a good news front is, my lab's not the only one developing RET degraders right now. There are actually some publications that just came out in the last 6 to 9 months from pharma companies like Bristol Myers Squibb that have actually developed some RET degraders to show proof of concept. So my guess is they're building some in-house, and we may hear about those in the next year or two and how those may work. So it's a pretty exciting time in that area.
This is very exciting, Dr. Drake, and I'm going to sort of regurgitate what I heard from you in terms of protein degradation, and please correct me if I'm wrong. So essentially what your lab's trying to do is hijack this natural machinery that all cells have. Let's call it, the cell's trash can where proteins go to get degraded. So your lab's coming in, creating these really, really cool molecules that take the RET protein to that trash can, essentially, and make sure that the protein goes away. Is that accurate?
Yes. Yeah. I mean, in a 35,000-foot view, that's very accurate. So yeah, we're trying to find that protein that's driving the cancer, and we're trying to chop it up into little pieces and throw it in the trash. Yep, exactly. That's essentially what we're trying to do.
And I love the fact that we're actually harnessing something that already exists in cancer cells, not trying to create something artificial, for lack of better words. So Dr. Drake, why are these potentially more advantageous than the tyrosine kinase inhibitors that we are using right now?
Yeah, so I think there's a few distinct advantages, and there are companies out there that are developing degraders for other contexts. So Arvinas is a company that's doing this for both the androgen receptor for prostate cancer and the estrogen receptor for breast cancer. That's where they've started. So the proof of concept is out there that degraders can potentially work and have some efficacy. So what we think a few advantages to degraders over kinase inhibitors is one, it has a longer lasting effect. So since it has a longer lasting effect, we can use 2 lower doses of it. And then if we can use lower doses, 3, it should be in theory less toxic to the patient. All right. So there should be less side effects. So those are like 3 big advantages that we think degraders can provide over kinase inhibitors. So what do you need to develop a degrader? Well, you need a compound that can bind to your target. All right.
And in
So
this
what you
case,
can't,
selpercatinib.
this could be selpercatinib, this could be pralsetinib, this could be a preclinical compound, as long as you know that it binds to, your target of interest, in this case, RET, and it doesn't bind to something else. All right. That would be, non-specific, which could cause toxicity in the patient. That's step 1. And then step 2 is then you have to link it to an E3 ubiquitin ligase, which is what I mentioned before. So there's really 3 components: the bind to RET, the linker, and the ligase. So you make those 3 components, you work out, so it's a lot of medicinal chemistry there, which I will not go into. We do have a partner that helps us with this as part of this award. And, then we can test it in cell lines first, and we can move to mouse models, and eventually we can, move to patients if all of that proves promising.
Very exciting, Dr. Drake. Question. You mentioned that all you need is a molecule that binds to your target, and in this case, it's RET and selpercatinib. Does that mean, and you also mentioned breast, the estrogen receptor, and prostate, the androgen receptor, but let's sort of go a little lung specific for a second. Do you think that this approach could also work for some of the other oncogenes that are seen in lung cancer, for example, ALK, EGFR, ROS1?
Yep, absolutely. It's theoretical because it hasn't been done, but I would say yes. In theory, you could absolutely go after those targets.
And Emily, I'm going to go back to a point you raised earlier on, which is the duration of the response, right? And Dr. Drake just mentioned that the technology seems to suggest that the effect of these drugs will last for a long time. What does that make you feel? How excited are you?
Incredibly excited. I cannot overstate how that makes me feel and how much I love the trash can metaphor for RET. I love that. I'm going to start using that. But I think duration of response is everything. We know that we do not yet have a cure for metastatic lung cancer or other cancers. But I think time is so precious, and when you have disease or your loved one has cancer, you are acutely mindful of that. So duration of response is not merely like data on a page, it's human lives.
Thank you, Emily, for that reminder. And the other piece, Dr. Drake, you mentioned is the ability to probably use smaller doses of such a very potent compound, which then leading to lesser side effects potentially for people with cancer. I think that's all great, right? Because people are living with cancer for longer periods of time and on these drugs for longer periods of time. So I think quality of life is equally important for our community. So again, thank you for everything that you're doing. And we have another question from the audience, which is focused on how these degraders work. Can you tell us a little bit about what that looks like? How do they cause cell death?
Yeah, so basically that's what that other component is of the degrader. So as I mentioned before, there's 3 pieces. All right, there's something that has to bind to your target. Then you have to add what's known as, we call them linkers. All right, so basically they're just chains of carbons. And then you have your E3 ubiquitin ligase on the other end. Okay, so that E3 ubiquitin ligase is really what does all the dirty work for you to eliminate that protein. All right, so that E3 ubiquitin ligase puts ubiquitin onto the RET gene, or onto the RET protein in this case. And when a protein has ubiquitin on it, that is when it gets targeted to the trash can. So basically it'll go through the proteasome, which is this several functional units that basically takes this protein RET and chops it up into little pieces. And then those little pieces get recycled and are reused for other purposes in the cell. All right, because they're broken down from
so there's a
proteins into
recycling
amino
component
acids.
as
Oh
well.
yeah, they get recycled. Yeah, yeah, it's very efficient, right? So all those amino acids that are chopped up, they get recycled and make new proteins, all right? They don't make RET, they'll make something else, right? That the cell might need. Or, the cell will die and it'll get eaten up by a macrophage, which is an immune cell, and that macrophage will recycle all those to a healthier cell. So that's typically, in a nutshell, how we think these degraders are going to work in that context.
Dr. Drake, since we're talking about the mechanism of action, right now this is all very preclinical, right? So your project is at the preclinical stage, which is obviously the foundation of all drug development. Can you sort of outline, what the next steps would be before this could be tested in a mouse model and then potentially moving to a clinical trial? What would those steps be?
Yeah, well, step one was to kind of get this concept moving in the first place. So that's where I can't thank LUNGevity and RET-positive enough for supporting this application. And we're moving forward with it, and it's greatly appreciated. So first, I want to say thank you, to you both for that, and to the RET-positive community for the support. So right now, there's several steps. So first, you have to generate a compound, all right, or degrader. And then you got to test the degrader on cell lines. So this would be what's known as in vitro work. So we can grow cells on a dish that have the RET fusion or have a RET mutation, or we can even artificially create these in cells.
Mm-hmm.
And we test to see, does this compound actually degrade RET? Does it eliminate the RET protein? All right. If the answer is yes, then we can move forward and start to think about, okay, this compound is promising. What's the next phase after that? Well, the next phase would be to ask, okay, go to the medicinal chemist and ask them, is this a formulation that would be amenable to something that could be taken orally? Because that's typically how you would administer this drug. So we would change the formulation up to see if we can make it an oral based formulation, and then go back into the cell lines and just confirm that it's still degrading RET as we think. Once all that happens, then we can move into animal studies. And the animal studies would test a couple things. One is, can we grow a tumor in a mouse, give the compound, and show we can degrade RET, and assess toxicity? All right, so those are kind of the four big things. So grow a tumor, oral administration, assess on-target effects to make sure we degrade the protein we think we're degrading, and then does the drug toxic or not? So if it passes all of those tests, then we typically move into another mammalian species to confirm it one more time. Sometimes this is in monkeys, sometimes this is in rabbits. I think it just depends on the study that you're trying to do before you would move into like a phase 1, phase 0 clinical trial to assess, toxicity and dose tolerability in patients. So those are kind of the steps that you would take to get from where we are today to the clinic.
And Dr. Drake, thank you again for reminding us how important it is to fund basic science. I think we often forget that this is where new ideas begin at the bench. Going back to sort of the mechanism again, and a question from the audience, would the degrader reduce the levels of the abnormal RET protein to normal levels? Is that a possibility, or is the goal really to eliminate the cell?
Yeah, the goal is to completely wipe it out, because eventually the goal is to kill that cell, right? You don't want that cell around. So that's the really goal, is to completely eliminate it. So, as with, the kinase inhibitors, those have off-target effects as well, and there's some toxicity associated with it. So, I would predict that these degraders will have some toxicity. We don't know what that is because we haven't tested it yet in the clinic, but yeah, the goal would be to completely eliminate that protein.
Love hearing that. Complete elimination. And Dr. Drake, this is a question that I had just hearing your answer, which is you talked about potential toxicities, right, of these compounds, but you also mentioned that, using a drug like selpercatinib, which is very specific to the kinase domain, is there a potential to sort of tweak the drug in a way that it only sort of attaches to some mutant part of the protein and spare the normal RET protein that's found in other cells of the body?
Yeah, that's a great question. I mean, so for RET fusions, all right, so these are a part, this is one version of a genomic alteration that can happen. What typically happens in a RET fusion positive tumor is that one half of the RET protein is intact and the other half is chopped off and is gone, and it fuses to something else that drives that RET activity. Okay. So this could be KIF5B, this could be something else that binds to it. That other half of RET that remains is still somewhat wild type RET. In other words, the sequence is normal. It's compared to a normal RET in a healthy cell. It's the same exact sequence. It just has higher function. It has higher activity. It's driving biology at a much quicker rate than you would expect it to do, which then leads to that overgrowth in that cancerous tumor, right? So in that context, in a RET fusion, it's still the normal sequence. All right.
Mm-hmm.
In tumors that are driven by RET mutations, then it's different, right? So you have an alteration of amino acid or two in the protein that then drives the function of that RET in the same way a fusion would, but it requires that RET protein to be abnormal sequence, has a different sequence than the healthy cells. So from that perspective, you don't necessarily need to design a compound that looks for the RET mutations, because it could bind to that RET fusion part and bind just as well. All right. And you also have to keep in mind, where does these drugs bind on the RET protein? They typically bind in what's known as the kinase domain of the protein.
Sure.
All right. And if there's no mutations there, even in the RET mutant tumors, typically those mutations are not in the kinase domain, they're somewhere else in the RET protein, then those inhibitors would work in both contexts. Now, where it gets tricky is if you have a treatment resistant tumor that developed RET solvent front mutations, that could then, render potentially that these compounds may not be as effective and may have reduced binding. And in that case, you may have to rethink about, what binder you'd want to use to bind to that RET protein itself. So that's something that we're also thinking about. When we test these degraders in our lab, we're testing them not only to the wild type or normal sequence of RET, but also to a bunch of the different RET mutations that may be seen in the clinic to confirm binding.
That's a great point, Dr. Drake, and thank you for sort of proactively testing some of these mutations and alterations. Again, sort of staying ahead of cancer cells, for lack of better words. Emily, you heard a lot of this discussion. I'd like you to tell us a little bit about what you think, or what else should the research community be focusing on, from a patient perspective.
Well, again, as I said already, I'm so excited about the potential for these protein inhibitors. I can't talk today. I love the potential for benefiting, cancer patients for a more extended period, of course. And but because I do think you asked me, Upal, the other day about what my greatest concern as a RET patient is for, our community. And that is when resistance develops to current drugs, our current inhibitors. And I think beyond that, we don't currently out of clinic have a great set of options. And so I think, this grant is, why we chose this grant, to meet that great unmet need, because for most of us, progression is inevitable. Now, it might be years down the road. We have people in our community who have been on selpercatinib or pralsetinib since before they were FDA approved, so for more than 5 years. But I think that, I know that immunotherapy, what is currently FDA approved does not work well for the large majority of RET patients. So that is something, I'm also very interested in, the longer term potential of harnessing our immune systems. But I think that what we're focusing on with Dr. Drake's work and research is incredibly exciting and really offers this additional time because, hopefully we all hope, and I believe if research continues at the pace it is, and that's an if, we are getting so close to so many treatments and, prolonging survival and, in some sense, you don't need a cure technically if you can live for a very long time. In the meantime, science is marching on all around you. But I'm rambling. But I would say, you know, these protein inhibitors, and I think also about immunotherapy advancements are the big things that I'm excited about.
You actually made a very beautiful point, my friend, which is about how when science is progressing, there's that next treatment option, the next treatment option.
Yes.
Even if we don't have a cure in sight, but if we have the next option that can get us closer to a cure, I think that's where science comes in. Dr. Drake, by the way, people watching in, we have a very, very enthusiastic audience, and people are asking about whether you have cell lines available for your work. And if not, people in Minnesota are offering their cells if you need cell lines to do your in vitro and in vivo work. Just putting that out there.
Oh, well, that's very, very generous. I mean, we do. So, and the beauty of what we can do with molecular biology is we can create these mutations and put them in cell lines that don't normally have it in the first place to test some of this stuff before we would actually use something where you see it naturally occurring in the cell, or what we call endogenous. But yeah, I mean, that's, very generous of your audience for sure. So,
Dr. Drake, question that I have, we talked about toxicity, we talked about coverage and being broad. But another question that I think is top of mind for our community is also if a drug enters the brain, if it can stop brain metastases or treat brain metastases. So do such degraders have the potential to enter the brain?
That's a good question. Don't know. There really haven't been any that I know of that have been tested for brain tumors in particular. So I think that's an unanswered question that we would have to find out, I think, when we were to move it to the clinic. So I think that's an unanswered question at this point.
And that's why we do research.
Yes. Yeah.
Dr. Drake, and going back to my original question about the steps, and again, from the audience, what's the timeline of taking, something like this to the clinic? Right now, you're doing all of the medicinal chemistry work, tweaking your compounds, then the mouse models, and then going to a phase 1. What could that look like in an ideal sort of situation?
Yeah, I mean, really one of the rate limiting steps in all of this is getting the right compound made. I mean, that just takes time. The chemistry has to work out perfectly to get the right yields and the purity that you need to then go on and test it. So once you get the chemistry figured out, the experiments that my lab would do would go fairly fast. The in vitro work in cell lines could happen very quickly. The in vivo work could happen very quickly. And at that point, it just becomes more of a scalable problem than anything else, right? So, in a perfect world, if we could get a compound, sometime in the year 2025, I would say by the end of 2026, we could be ready to really consider whether this compound is something that we could start in a phase 0 in 2027 at some point. That's in a perfect situation. But I think it could happen that quickly if we get the right compound in place.
And let's stick to the perfect situation because we want to be hopeful and optimistic. And with that, Dr. Drake, I'm going to ask you to be a little philosophical, and let's talk just about the role of preclinical research. And, as a PhD scientist, you focus on preclinical research. Why is that important? Because again, sometimes on the patient advocacy side, we focus only on clinical trials, which is really the lifeline of patients. But I think it starts much before, right? So from your perspective as a leader in preclinical research, as what your lab focuses on, why is it important and why should we be funding it?
Yeah, well, without preclinical research, you can't develop drugs and you can't have clinical trials in the first place. So, the general goal of preclinical research is to help generate knowledge about mechanisms and interactions of proteins and pathways to understand disease. I mean, that's really what we try to do as a basic scientist. Now, my lab's more translational. We try to take what we learn and try to move it towards the clinic, right? But I don't treat patients. I don't see patients because I'm not a treating physician. I'm a PhD. So from that perspective, preclinical research is very important because if you can't understand mechanisms, it's going to be hard to develop drugs against whatever target that you're looking at. And to be honest, it is a healthy relationship between what we do in our labs and with industry, because industry are the ones that actually develop the drugs, you know, and a lot of that is brought on through funding through the NIH and through the Department of Defense. And, you know, if industry doesn't understand mechanisms, they're not going to be able to develop the drug because they don't know what they're developing the drug against. So they take what we do and they build on it to commercialize and to build products for patients with disease. So I think that's really the main reason why preclinical research is very important, and we should always consider preclinical research as a starting point to help us understand. And the stuff that you see in the clinical trials is kind of more the end point. You know, we've understood something, now we're going to go test it and see, does it actually, you know, hold true in a patient setting. And that's, you know, that's the value that preclinical research can bring.
That was the best description of preclinical research and the research ecosystem, Dr. Drake. And I'll tell you why I love it is because you reminded all of us how researchers such as yourselves, who are funded through the NIH or the Department of Defense, work very closely, and the knowledge that you generate impacts drug development with pharma, and how this ecosystem is so well oiled. And of course, with clinicians, you know, basic scientists such as yourself, clinicians and pharma work so closely together to make sure that these innovative ideas really reach patients. And I think we sometimes forget that it's this partnership, this ecosystem that makes all of this magic work. And I think, you know, we are almost at time. So Emily, my friend, I'm going to ask you to bring us home with a message for our community. And when I say our community, what's the message you have for advocates, for anyone who's listening in, or policymakers?
Sure, my pleasure to take this on. I will just say as concisely as I am able, a little bit very briefly about NIH, National Institutes of Health. They are the world's largest biomedical funder, and it's not even close. It's like 34 times, I think, the figure that the next highest funder of research, that's in the UK, invests in this research. And we are going to be publishing our RET-positive newsletter next week, Dolores and I are, and we will include some nice graphs and some other information just so you all have, you know, the picture of what federally funded research makes possible. And the United States has been the global leader in biomedical research, treatment development. I can tell you as a board member for RET-positive, we get emails almost every month through our website from people in other countries who do not have access to the drugs we do in the United States. And we know we have a community beyond the US. And but we are really at the global pinnacle of this sort of research. And we have really been in a golden age in the last, I think, 15, 20 years in terms of drug development. So all lung cancer drugs, just a few pieces of information for everyone. All lung cancer drugs are approved in the last 15 years, have been supported by NIH funding directly or indirectly. And so that's what Dr. Drake is talking about, look, the preclinical foundational science. And I think that's especially important for rare oncogenes like RET. You know, Lilly Pharma, who makes Retevmo, probably would never have invested in this amazing molecule that treats disease for such a rare oncogene without federally funded research. So it's very important. And, you know, where we are at present, I will just say as a lawyer, is a very precarious place in terms of federal funding in the United States, both NIH funding and, as Upal says, the Department of Defense funding. So in terms of NIH funding, and 83% of NIH funding last year went to grants to researchers like Dr. Drake, who don't work for NIH directly, but work across the country in labs and universities across the country. So we are really reliant on people like Dr. Drake, and so grateful for you and the work you do for us. I would just say, in terms of the NIH funding concerns, the executive branch issued a 15% indirect cost cap back at the very beginning of February. I will say, putting my lawyer hat back on, this is not constitutional. You know, we have 3 separate branches of the government: executive, legislative, and judicial, and only Congress can constitutionally appropriate funding or make any changes to funding. So under federal law, you know, if you want to make any changes to the NIH funding policies, that has to be done through Congress. So where we are in terms of the legal issues is their 22 state attorneys general have filed a lawsuit over this 15% cap, along with multiple universities and research institutions. And a federal judge in Massachusetts has issued a temporary restraining order. So I think that this is my personal, obviously my hope, all of our hopes, but my feeling is this will be in time resolved in terms of the funding cap. You know, under federal law, which is the current law and existing law, universities negotiate those indirect costs with NIH directly. So that goes for things like very expensive lab equipment, support staff, keeping the lights on, keeping these labs in continuous, stable operation, because the last thing that we need Dr. Drake worrying about is whether he's going to be open in 6 months. So that continuity is so critical to this scientific discovery. The other thing, big thing that I think that is going on at present with federally funded research is what Upal mentioned. That is the Department of Defense funded research. And 2 weeks ago, Congress voted to cut that funding by 57%. And I can tell you all that my doctor, Jared Weiss at UNC, his research is funded by the Department of Defense research so that is a major blow to cancer research across the board. And what I would say, getting to your question, Upal, in terms of what we can do, you know, I think when I was first diagnosed, I think one of the first realizations I had in terms of my really healing and starting to stabilize was this realization that I'm not the first person to get cancer. I'm not the first person in my 30s to get cancer. And I'm not alone. And finding RET-positive and being part of our patient, caregiver, research, advocacy community across the board is, I think, one of the most important things any of us who really care about, who have a loved one, who ourselves have this horrific, fatal disease, is be an advocate for our community. And in doing that, we advocate for research funding. So like I said, our newsletter is going to come out next week. We will have some information about kind of the general state of things and also some ways in which you can get involved. You can contact your legislators. You know, I think that it is a bipartisan issue, cancer research. No one wants cancer patients to not have the treatment we need to stay alive. So I think it's a very sympathetic and powerful issue. But I think that the general public really doesn't know what the role of federal funds play, what they play in our research and what they play in terms of industry and like big pharmaceutical for-profit companies developing this amazing science that is first founded, you know, the foundation is laid by researchers at universities and other institutions. So speaking out, advocating, and we will always let you know in the RET-positive and broader lung cancer community about things that are happening, things you need to contact your lawmakers about. So I think that's the biggest thing right now at this time that we need to be doing as advocates and patients and caregivers.
Emily, I could have not summarized it so beautifully. Thank you for reminding us that cancer is not political. Science should not be political. And at the end of the day, science is here to really improve the outcomes of people living with certain diseases. And I think we just need to keep that in mind. And I really appreciate also reminding us how we can take action and and unite. And last but not least, I think the importance of your funding is so critical right now when science is where federal funding is a little precarious and decisions and situations are changing. I think your support makes even more of a difference. So thank you to you, to Emily, to Hillary, to the RET-positive community for your advocacy, for your community building, and for your support of research, my friend. And with that, we can wrap up. But before that, I'm going to just spring this on both of you. People loved hearing this discussion. So by popular demand, you guys have to be back in a couple of months with an update.
Okay.
Happy to join whenever. So I had a great time.
Likewise.
Thank you so much again, Dr. Drake and Emily, for joining us. And thank you to our attendees for listening in, for your amazing questions.
All right. Well, thank you very much. It was great to see you, Upal and Emily, and give my best to Hillary as well. I wish she could have made it, but and thanks to the RET-positive community and LUNGevity for funding our work. And we will do our best to bring new products to treat this disease.
Thank you again, Dr. Drake, for your passion and commitment.
All right. Well, take care, everyone. Have a great weekend.
Take care. Bye-bye.